It's my pleasure to introduce our speaker, and that's Dr. Brad Nelson. And he works at the BC Cancer Agency right here in Victoria. He's a distinguished scientist and director of the Dealey Research Center. And he's the co-director of the Immunotherapy program. I'm struggling with some of these words. He's a native of Vancouver. He has a Bachelor of Science from UBC in 1987. He has a PhD from the University of California at Berkeley from 1991. He has a postdoctoral training with Dr. Philip Greenberg and has held faculty positions at the Fred Hutchinson Cancer Center and the University of Washington in Seattle. In 2003, he became the founding director of the BC Cancer Agency's Dealey Research Center here in Victoria. He's a professor of medical genetics at UBC and a professor of biochemistry and microbiology at the University of Victoria. Dr. Nelson's lab uses genomic, how do we say it? Genomic, okay, and molecular approaches to study the immune responses to cancer with an emphasis on ovarian cancer. As the co-director of the BC Cancer Agency's Immunotherapy program, he is leading a phase 1 clinical trial program focused on adoptive T-cell therapy for gynecological cancer, leukemia, lymphoma, and other malignancies. In the last week, I actually saw an announcement which read, "Game-changing therapies, one step closer at the Cancer Agency." And right away I was interested. New information, new treatments possibly coming down the pipe. So I read that Dr. Francis Bernard and Brad Nelson are the recipients of close to $8 million in grants from the Canadian Foundation in Innovation. And they're there to develop novel therapies using rare isotopes and immune therapy respectively. So that's really good news for us. Congratulations. So Dr. Nelson's topic tonight is immunotherapy, a revolution in cancer treatments. Please join me in welcoming him.
Well, thanks very much for the invitation to be here tonight and for that warm introduction. And I was just noticing as I was staring at my own slides, Executive Resource Group, this is a very rare day where I actually gave 2 presentations in the same day. I'll admit that. And this was a group I met with at lunch today. But I'm here to talk about immunotherapy. You may or may not know what that is, but about 40 minutes from now, hopefully you'll have some understanding of what I'm talking about and why it really is a revolution in cancer treatment. I'm going to start by saying, recognizing this is a prostate group, immunotherapy has yet to have a big impact on prostate cancer. But as we go through this, I'll explain why, and I'll also explain why we think it's going to have a major impact in the very near future. So we're not there yet, but there's huge promise. So I just want to say that right off the bat because I know what your primary interest is here. And before I go on, can anyone tell me where this picture was taken?
West Coast somewhere.
So Vancouver Island somewhere.
Your group?
Pardon me?
Your group?
No, it's Hot Springs Cove up near Tofino. If you've not been there, it's a beautiful, beautiful spot with natural outdoor hot springs. Great, and I work just across the parking lot at the Cancer Center. Probably many of you have been there, unwillingly perhaps, but I hope that you've well there have received good treatment and have come to appreciate what we're able to do at the Cancer Agency with today's treatments in prostate cancer, as you'd be well aware, is one of the cancers where we've had the most success so far with research and treatment, and overall very high survival rates compared to other cancers. And as I said, as we go through the evening, I'll talk about how immunotherapy is going to make that even better as we go forward. So if you've been to the clinic, you would have been on the first or the second floors where the treatments are given, and maybe you've come up to the third floor for the nutrition counseling or other forms of counseling, and various education classes that are held up there. Our research center is, I'll just point to it, if you haven't been, it actually occupies this wing on the third floor. So it's the Dealey Research Center right up at the top of the stairs. And before I forget to mention it, we do monthly tours of the research center. So if you're left with an appetite to know more after tonight, let me know and I can get you on the mailing list for those, so that you can come and see things up close and personal and ask a few more questions perhaps. So the topics I want to cover briefly, but is to get warmed up, we'll talk just briefly about what cancer is, how I see it as a scientist. We'll talk about what the immune system is, how does the immune system respond to cancer, and why is immunotherapy now the hottest field in oncology, and what are we doing about it locally and nationally in this field. So I'm going to cover all these topics. So let's just get warmed up with what cancer is and how it develops, because that just helps give a framework for everything we're going to talk about. So cancer starts, and I hope you can you still hear me if I get away from that microphone? Maybe this one will pick me up. I like to point. But we're starting over here with just a normal healthy tissue. This could be your skin, the lining of your lung, your prostate, wherever it may be, but just some healthy cells forming a healthy tissue. Cancer starts one day when just one of those cells suffers some sort of genetic damage that actually changes the programming in the cell. If it was your laptop, it would be like changing the operating system, changing a few of the bits in your operating system. So now the cell starts behaving in a different way. Now, just like your laptop, if you change the code in it, it would probably crash. It's probably most of the time would be the result. And so most would-be cancer cells probably die off as well. These mutations generally are bad. But once in a blue moon, all the stars align in a bad way, and cancer cells can end up with just the perfect combination of mutations that, instead of killing the cell off, actually gives it super growth powers. So it can start just dividing uncontrollably. And that then leads to something we call dysplasia, or just a benign lump. We've all got probably hundreds of these in our body, and we don't have to worry about them. Most of the time they just stay put at that point. But if more bad luck comes along, the cancer can then progress to an early stage cancer, which we call an in situ carcinoma. And then if that's not dealt with, it can go to become an invasive cancer, meaning that it starts to actually invade the tissue around it and starts spreading to other parts of the body. And that's when cancer becomes the most dangerous and the hardest to treat. So a surgeon, and often with prostate cancer, this would be the approach, can come in and cut out these early stage cancers, and if they get it all, that's curative. Nothing left, no problem left behind. It's when the cancers start spreading to other parts of the body, though, as you can imagine, no matter how good a surgeon is, they can only cut out what they can see and what they know about. And if there are, small deposits of cancer hiding from imaging, then surgery is not going to do it. And so that's when we need things like hormone therapy, of course, in the case of prostate cancer, chemotherapy for other cancers. And it's also where immunotherapy is going to have the biggest impact, as I'll show you. So overlaying on all this, if you're looking inside the cells, what we're seeing is over time, these cancer cells are getting more and more mutations. They're becoming more and more abnormal. And they're actually undergoing a form of Darwinian selection, meaning that they're sort of evolving much like animals have evolved on the earth. And so the cancer you start out with actually starts to change over time. And that's what makes cancer also difficult to deal with, because it's because of this change. So you can come in with a treatment that might kill 99% of the cancer cells, but the 1% potentially can then be selected for and grow out and form a new tumor. So these are the challenges we face, and I'm sure you know this all too well, having those of you that have gone through this journey. But there is some good news in all this. Every story with villains also has some heroes, and can't get better than Charlie's Angels. So who are the heroes in this story? The heroes are the immune system. So what is the immune system then? Well, it's not a skeleton. That's just the starting point. The immune system, we're going to talk about your bloodstream. It's basically what's going on in your bloodstream. So you've got your arteries, which are carrying blood and oxygen and all that good stuff throughout your body. You've got your veins, shown here in blue, which are sort of draining, bringing the blood back to the heart ultimately to complete that circulatory loop. And then you've got another system of plumbing called the lymphatic system, and that's colored in green now. It's sort of a third set of, as I say, plumbing or vessels that runs through our body. And so we end up having this really sophisticated network of blood vessels and lymphatic vessels reaching to every corner of our body. And this is important because we need the blood to get to every cell in our body to deliver oxygen, or we wouldn't live very long without that. But as I'll talk about, this same system is used to deliver our infection-fighting immune cells. And so coursing through these various vessels is blood. And I'll just blow this up so we can see what's in blood. So blood has what most people are most familiar with are your red blood cells. These are these disc-like cells, and they are the ones that carry oxygen, and they're the ones that make your blood look red. So we like these guys. In fact, we like everything in your blood. But the white blood cells are these other types, and there's a bunch of different categories, macrophages, neutrophils, etc. And we're going to talk about a particular white blood cell called your lymphocytes, and even narrower than that, T cells in particular. So these white blood cells travel around your body to every nook and cranny, head to toe, 1 million miles an hour every day, and they're looking for any signs of infection or, as we'll talk about, cancer. They can also recognize and start attacking cancer. And that's what keeps us healthy most of the time. So as I said, we're going to narrow in even further now from white blood cells to a particular type of white blood cell called T cells. And this is a sort of artist rendition of what T cells look like in sort of a generic diseased cell. This could be a cancer cell. This could be a cell that's infected with a virus, various types of disease. And T cells are the good guys. A few of them are shown here. And they have the ability, quite literally as shown here, to reach out, and they kind of crawl around the body and they feel other cells. And through various chemical sensing pathways, they can tell whether a cell is diseased or not. We won't get too far into the details, but there's very sophisticated pattern recognition mechanisms that are used by the immune system to do this. And if this T cell decides that this is a diseased cell, what it'll do is it will literally then come right up to it and stick onto it, poke holes in the membrane, inject toxic compounds inside the cell, and the diseased cell will just dissolve within a few minutes. It'll just be digested away. And that's how the immune system deals with if a got a virus infection, let's say in your throat, and your throat cells have a virus in them, how do you get that virus out of the cell? You don't. You actually just kill all the cells that have the virus in them. And then those will then be replaced by healthy tissue a few days or weeks later. So that's how T cells work. And to show how important they are, when we talk about AIDS patients, for example, people with HIV infection, where what gets damaged there are your T cells. They actually start getting attacked by the virus. They disappear. And then we can see all the awful diseases that then unfold when you don't have T cells protecting you. So it turns out, as I mentioned, T cells can also recognize cancer cells. So this is a picture, it might look like a cartoon, but this is a real cancer from a patient with ovarian cancer. And we're looking inside her tumor, and all these sort of bluey purple cells, those are the cancer cells, disease cells. And if we look in particular at what we call the killer T cells, these brown cells, and they're not normally brown, we just colored them to look this way. But you can see, just like that cartoon I showed you, they're actually reaching out and touching the cells around them and trying to make a decision whether they should kill that cell or not. Helper T cells are in there. They provide a helping role. B cells are another type of immune cell that also helps. So it's really a team of good guys, like Charlie's Angels, that come in and work together to deal with these situations. So this patient, like others like her, this is what her immune system is naturally doing to fight her cancer. We didn't do anything to make these immune responses happen. This is, she's essentially walked in off the street for her first surgery, but we can then get a sample of the tumor and look and see that her immune system is already doing its best to try and fight this thing. But the woman still has cancer, so obviously something's not working optimally. So I'm now going to expand this a little bit more by, if we look at tumors now for multiple patients, we can see there's actually quite a diversity of these responses. So I'm showing you samples from 3 different patients, all with ovarian cancer. And this patient is one like the one we were just looking at. The colors have changed, but that's just a technical thing. But the point is, she's got a really strong immune response. But we see patients that are somewhere in the middle. They've got a weak or moderate immune response. And other patients, we look in their tumor and there's nothing going on. There is no immune response. These are all just cancer cells. And if we look at hundreds of patients and we categorize them this way and then follow them over time, we can find that if you've got a good, strong immune response underway, you've got about a 3 times better chance of surviving. This is for ovarian cancer, than somebody who has no immune response going on. And the same is true for prostate cancer, colon cancer, lung cancer. When we see these immune responses, it's a good sign. And it's not perfect, but it's an indicator that you're more likely to do well. So we've known this for a couple of decades now. And with immunotherapy, the title of my talk, what we're trying to do is strengthen these responses even more. Because this person still has cancer. So this is good, but it's not good enough. And her survival is going to be about 50% likelihood, but we want it to be 100%, of course. And we also have to think about patients like this, where their immune system isn't even getting started. And how can we turn on those responses and get them ramped up to this level and beyond? So with immunotherapy, which I'll describe how we do this, but that's the objective, is just to take what's already starting to happen and make it that much stronger. So the question is, let's start with the best case scenario here, this patient. Why does she have cancer? Why are these T cells not being successful at eliminating this tumor? Well, this is where the first breakthrough comes. And I'll explain a little bit of the background about this. But a lot of really smart scientists over the last 20 years have been studying how T cells work. How do they recognize a diseased cell and tell the difference between that and a healthy cell? And the first thing we've learned is there's, shown in orange here, there's a receptor that can sort of reach out and look for signs of disease or damage in the tumor cell. That's step one. And we can think of that as the gas pedal. If this receptor is being activated, it tells the T cell it should be driving up to this tumor cell and thinking about killing it. But then we've got another receptor shown here, and it's got the name PD1. Doesn't matter what that stands for, but it's another receptor that serves as a brake. And this actually sends a message to the T cell to stop. So the T cell's getting a bit confused now. It's on the one hand being told kill, and on the other hand being told don't kill. And why would the system be this way? Well, T cells are kind of dangerous things. We don't want them attacking our healthy tissues, or else we'd have autoimmunity, or inflammatory bowel disease, or allergies, the things that you hear about that are caused by the immune system. So Mother Nature has endowed us with a lot of brakes in our immune system as well, to make sure that the things stay under control most of the time. And the problem with cancer is those brakes are getting in the way. So this is what this looks like in the real world. This is, again, an ovarian cancer. Stained colors have changed once again, but the good guys, the T cells, are these green cells. And the bad guys are these other cells, macrophages, which are expressing this brake. And you can see for yourself, pretty much everywhere there's a T cell that's trying to kill the tumor, there's a cloud of these braking macrophages around it, stopping it from doing that. So you can think of it as a stalemate, and the response is being held back. You can also think of it like this. It's like being in a car with the driver from hell who has their foot on the gas and on the brake at the same time. And you're maybe stuttering forward ever so slowly, but mostly you're just getting frustrated. So that's what's going on in these tumors.
Excuse me, is that an actual image from a cancer? That's actually the cells that you're looking at?
Yeah, these are real cells in a real tumor.
And how do you get them colored like that?
We use a method called immunohistochemistry, using antibodies and colored dyes. We can, depending how we set it up, we can sort of light up whichever cells we're interested in seeing.
Terrific.
So if you come for a tour of the lab, we can show you exactly how we do that. This is not a real picture. For this, we go to Google Images. Okay, but so all these smart people have been working on this problem, and it's really paid off, because once we, and I wasn't the smart person that figured this out, but once the braking mechanism was understood, people came up with drugs that would interrupt that brake. So antibodies that actually just go and glom onto the brake so that you can no longer make this contact. And so now you've got a situation where the T cell is getting its foot on the gas, and so it's getting the go signal, and it's no longer getting the stop signal. And now it can charge ahead and actually kill that tumor cell. So that whole class of drugs are called, for various reasons, they become known as checkpoint inhibitors. And this is the first of two big breakthroughs I'm going to tell you about. So some of the drugs in this class are ipilimumab, nivolumab, etc. And just by giving these alone, you can see, first of all the different types of cancer where they're showing effectiveness. And in blue, I'm indicating all the FDA approvals that have occurred. And so that's the Food and Drug Administration in the US. and this is sort of an international standard. When something achieves FDA approval, it means it's actually working. There's hard, concrete scientific evidence that this is actually working. Now, and the response rates in melanoma can be as high as 50% of patients are benefiting from these. In lung cancer, it's down around 20%. Not 100% is what we're after, of course, but 20% in this really terrible set of diseases that most other drugs have been very ineffective in. This has caused a lot of excitement. Kidney cancer, bladder cancer, Hodgkin's lymphoma, 87% response rate in an otherwise fatal lymphoma. And so this has all just happened in the last, you can see the dates as well, just in the last 4 or 5 years. And this is really unprecedented in oncology to have a class of drugs just sweep through the cancers like this. Prostate cancer is not here. Prostate cancer is not responding well to these drugs, and I'll say a bit more about that and what we need to do instead. Ovarian cancer, which I mostly study, also is not benefiting very well from this. But we're also, one other good thing to mention is we're even seeing for melanoma and lung cancer, these drugs are starting to replace chemotherapy as the frontline treatment because they work better and they have far fewer side effects than the chemo. So we're actually seeing an era now where chemo is actually being pushed out of the way in favor of an entirely new type of treatment. So, what's the downside? Well, cost. These are expensive drugs, about $100,000 treatment, which is on the sort of upper end of what oncology drugs cost. We need to hit a couple of the breaks, and so combinations may be required. That doubles the cost. It doubles the side effects for patients. Not optimal. And long-term use may be required. And in fact, that's the big question now in the world is how long do people need to stay on these drugs? The hope is that once you get the immune system working again, that maybe it'll just keep working and you can ease off on these drugs. But you can imagine there's reluctance to stop giving somebody a drug if it seems to be curing them. And as mentioned, many cancers, and I could add prostate to this list, have low response rates. And even where we're seeing successes, the responses often are transient, so that the cancer is coming back in a lot of cases. So lots to be excited about, but also a lot of work still to be done that lies ahead. So just to give you a feel for why, what needs to be done next, and why this is possibly not working in every type of cancer. I've shown you one brake and one gas pedal, but this is actually our current understanding of all the brakes and gas pedals that really exist on T cells. So these in red are all the different braking mechanisms, and I talked about PD1, but it's just one of many brakes in the system. And these are all the gas pedals, so likewise there's multiple signaling pathways here. So biology is always much more complicated than the human mind would like it to be. But this is the reality of how T cells recognize their targets. They use very sophisticated mechanisms. So to make an analogy, we'd like it perhaps to be as simple as a Model T with a brake and a gas pedal like I've been talking about. But in reality, T cells are more like 747s with very complex control mechanisms. And we should actually be thankful that they're this complicated, or else we probably wouldn't be on the planet. We would have been wiped out by some terrible virus or something eons ago. So we've got these really sophisticated T cells. Now the question is, how can we actually become the pilots, jump in these T cells, and actually fly them the way we think they should be flown and into the targets that we really want them to hit? How can we really understand this system to the point where we can drive things in the direction we want them to go? And so that leads to the second of two approaches I'm going to talk about, and where I think the hope really lies with prostate cancer. And this is an approach called adoptive T-cell therapy, or ACT. And the way it works, there's different flavors of it. I'll explain a couple different examples, but they all have the same basic principle. You start by collecting either a blood sample or a tumor sample from the patient, and you can then pull the T cells out of that. And you get them in a dish in the lab, and then you can identify the ones that recognize the tumor, or you can even, as I'll show you, you can actually engineer them, genetically engineer them to recognize the tumor. And so you do all that while they're in the lab, and then create basically an IV bag full of these really optimized T cells, and give them back to the patient, put them back into their bloodstream. And as I explained at the beginning, now they're in the bloodstream, so these T cells will go to all corners of the body, and they should be able to recognize and destroy the cancer cells wherever they might be hiding. So it's a way of really sort of amplifying these immune responses by about 1 million-fold. And I'm not exaggerating, it's sort of on that order of amplification that we can achieve. So this is what this looks like. I'll just show you, this is an example from a mouse experiment we did about 10 years ago now. We had mice that developed breast cancer, a mouse version of breast cancer. And this is what the tumors look like. If we don't treat the animals, what we're seeing here is pretty much all tumor cells, very few T cells. And here's what we can achieve just a week after doing this adoptive T cell therapy. All of these brown circles here, these are all T cells which have now flooded into the tumor and are attacking it. And in the mice, when we do this, within about 10 days of treatment, we can make these tumors completely disappear and never come back. We can have curative responses that way. So we've known this about mice, what we can do in mice for quite a while now, but the big challenge has been to get it working in people. So this is what we're gearing up to do right now. And we've built what we call the Conconi Family Immunotherapy Lab. Conconi family are shown here, various very generous donors to the BC Cancer Foundation put a lead gift of $2 million towards this project. And then thousands of other British Columbians pitched in, and we got a pot of about $6 million altogether to build this facility. You mentioned our recent Canada Foundation for Innovation Award. When we get our provincial match, that'll actually be a total award of $11 million that will add to this. So we've put together close to $20 million for this project, which is going to allow us, as I'll explain, to do a lot of really exciting things over the coming years. So we built this immunotherapy lab because to actually produce T cells that you're going to get back to patients, you'll be reassured to know that there's all sorts of rules about how we do that and standards we need to meet. Of course, the T cells have to be sterile, they have to have the right qualities, and Health Canada will be watching us very closely to make sure that we do this in the best possible way. So to achieve all those standards, you need what we call a clean room, and this is what our facility looks like. And basically, the approach we're using is a sort of a glove box design. This is a glass cabinet, and the operator here, you can see, is reaching in through these sterile gloves and doing all the manipulations in this way. So the T cells basically live in this perfect bubble and spend their whole time in there and never come in contact with the outside world, so that in that way they stay perfectly sterile and well controlled. And so if you come on a tour at the lab, can show you more and tell you more about how all this is done. But this facility just opened about a year ago, and we're just now operationalizing it and getting ready for our first clinical trials. So the first trial we're going to do is relatively simple, although it's taking us a few years to get it all in place, but the concept will be to take tumors like from these two types of patients, we can actually pull the T cells out of these tumors, and they're in there fighting the good battle, but they're losing, and there's not enough of them. So we'll take them out and expand them to larger numbers in the lab, and then give them back. So the way that'll look, this is a typical ovarian cancer patient, just kind of looking at her, what happens today, sadly, to so many women with this disease, is they go through multiple rounds of chemotherapy, and their tumor just keeps coming back, and ultimately many of them die from their cancer. What we'll be doing with our first trial is inviting women to participate when they have their first recurrence. We'll be able to collect the tumor at that point, pull these T cells out, and start growing them in the lab. The patients will go on and have their second line of chemo, which would still be part of the standard treatment. And then if they relapse a second time, instead of having more chemo, which we know is ultimately going to be futile, instead we'll be offering them an infusion of their own T cells that we've now given a little respite in the lab. We've fed them, rested them, given them new ammunition, and expanded their numbers, as I said, about 1 million-fold. And so the patients will get a fresh dose of T cells, which we hope will then eradicate this recurrent disease. So that's the plan for our first trial. And then we'll be opening, we hope, next spring. And I'm going to end by telling you what the future holds and where, and this is the treatment that I think ultimately holds promise for prostate cancer, is taking things yet another step further and actually talking about genetically engineering the T cells to ensure that they recognize the tumor. And there's no if, ands, or buts about it. So again, back to this picture, these T cells reaching out and trying to decide if this is a disease cell. We can engineer the T cells so that it has a perfect recognition of cancer, and without question will recognize an attack. And the way that's been done, and this is work that started in Israel and was then developed further in the United States, and we're bringing to Canada now, is a strategy called chimeric antigen receptors, which we just call CARs. And I won't get too into the details here, but this is the surface of the T cell, and what it's essentially doing is putting an antenna on these cells, and this part can actually grab onto the tumor cell and bind to it, and it's attached to this stem here, which sends an activating signal to the T cell, telling it to attack. And so the innovation here was to take part of an antibody and part of a T cell receptor and put them together and create this chimeric protein. So that was the genius of a group in Israel thought to do this. And the first CAR T-cell trials were published just about 4 years ago now. And the first trials were done in leukemia, and just absolutely mind-blowing results were achieved. So 90% complete response rates, of which about 2/3 are sustained. This is in pediatric and adult leukemias. So a complete response is just what you think it might mean. It means the tumor goes away and is undetectable by any means in these patients. So in 90% of patients, they're achieving this elimination of the cancer. Now, it comes back in about 1/3, and we'll talk about how to deal with that problem, but for 2/3, it seems to actually be curative. And these are really, really lousy leukemias that are resistant to every other type of treatment. These are patients who were out of options until CAR T-cells came along. So it's really been a game changer. And was just FDA approved in the last, one product in August and another in October just of this year. So this is really breaking news. So while I'm on this slide, I'll just point out, now that we know this basic design, it's really as simple as finding things on prostate cancer cells that the antibody can grab onto. And we can, in theory, engineer T cells to recognize any type of cancer now. So leukemia is the starting point, but all the other cancers are on everybody's hit list now to develop this strategy towards, including also ovarian cancer, which I said is an interest of mine. So again, what's the downside of all this? There's a lot of side effects that have come up. A lot of these now are manageable. Now that there's some clinical experience, doctors know what to look for, and they can deal with a lot of this. But still, it's considered, pretty high risk treatment, and you need to be admitted to the hospital to receive it. I'll talk about how we can improve on that. There's a few other things. There's relapse is an issue, as I mentioned, and cost once again comes up. So these treatments are being commercialized in the U.S., and the companies that are doing this are going to be charging between $373,000 and $475,000 just for the T cells. You still have to pay for all the clinical care costs of the patient, including the hospitalization costs. So if you put it all together, this treatment is going to cost upwards of $1 million per patient. So we've gone from thinking $100,000 treatment is pushing it to how do we even think about $1 million treatments? And yet, with a 90% success rate, how can you not think about these $1 million treatments? So I'm going to address that in a minute. But I think most importantly, this has to be seen as great news and a great breakthrough in medical science. And so, but now we can sit back and say, okay, but what's our wish list? What would the perfect strategy look like? If we could just blue sky it and a genie came and gave us whatever, 6 wishes, what would they be? So I won't go through these one by one, but there's a lot of engineering things we can think about. This initial CAR design already just looks, too simplistic. There's all sorts of switches and control mechanisms we could build into it, which would make it safer, get to the point where you don't need to go to the hospital to have it, so our care costs would go down, etc. Getting it to work against prostate cancer, of course, common epithelial cancers is a big goal. T cells sometimes have trouble getting into solid tumors. Leukemias are what we call liquid tumors. They're actually floating around in the blood. But T cells often don't like to go into a big lump. They kind of get stuck on the margins. So we need to deal with that. So there's a lot of work to be done. And I'll give you one slide on how we're going to approach that, and then I'll talk about the affordability issue. But in terms of just better engineering design, I'm working with a professor at UVic, Marty Boulanger, and he and I are working on something, an alternative to CARs, which we call MATES, or Mutual Antibody T-Cell Engagers. It's just an acronym, but what we've done, or the idea is, taking the basic CAR design and just breaking it into 2 pieces. So the antibody, the part that grabs onto the tumor, is actually a separate piece from the receptor that sends the signal. And then Marty is creating the equivalent of a Velcro-like mechanism. It's protein Velcro, so that these 2 pieces can actually stick together as if held by Velcro. So you end up back to where we started with basically a CAR again, but with this detachable part now. So the reason we want to do that is you could then give the patients the T cells expressing just this half, and the doctors would have to actually then give the patients this antibody, and they would do that as if it were a drug, and let the antibody find the T cells in the body and create a CAR. The advantage then is if there's side effects, if the reaction is too strong, you just give less of the antibody, and you can kind of turn the system down. Or if it's not working well enough, you just give more antibody and turn it up. If the patient's been cured and you no longer need these CAR T cells, just stop giving the antibody and everything should settle down. If the tumor evolves and the target is no longer present on the tumor, but there's a new target, you could give a different antibody. So it allows you to kind of switch your targets in real time. So we're excited about this, and we've got a grant from the Leukemia and Lymphoma Society to build the first generation of this. And I put it forward as just one example of a pretty simple idea that we think is going to actually resolve many of the issues with CAR T cells. So what about, yeah?
So those T cells that you've introduced, do they then naturally reproduce in your system?
Yeah, thanks for bringing that up. Yeah, so the T cells actually, we call them living drugs, because they actually start dividing when they're in the patient's body. So you actually can end up with more T cells than you're actually given. So I'll end just by thinking about the affordability and feasibility. And I just want to quote Tommy Douglas, who some of you may know is really credited for creating our Canadian healthcare system. And there's probably days when you might curse him for doing that, but I actually think we've got the truth is worldwide, we've got a fantastic healthcare system. Lots of we need to do to improve it, of course, but the fundamental idea being that, as he said, whatever health services a person requires, they should have access to, irrespective of their individual capacity to pay. So I think most of us as Canadians buy into this concept. And but how do we hang on to this value? How do we keep this promise when we're now in an era of $1 million treatments? Our cancer clinic across the way, we see 5,000 new patients every year, just here in Victoria. If every one of them ended up needing $1 million treatment, that would be $5 billion. You did the math, just for our little clinic. So these prices are not sustainable. So our view of how to deal with this is to create a new model that actually makes sense within our environment. So the current model, the commercial model, is the companies own this technology, and they're going to build factories that make these T cells. So Novartis is one of the companies involved, and I'm not picking on them. They do a lot of good things, but their model is you send your T cells to New Jersey, to their factory, and they come back a couple of weeks later, and they've now been engineered to have this CAR in them. And it comes with a bill for $475,000. We think what makes more sense is to do what we've always done with bone marrow and stem cell transplants, which is we don't ship the cells out of the country, but hospitals have trained experts that know how to do the cell processing right within the hospital. Here in BC, that's done in Vancouver at the bone marrow transplant unit for treating blood cancers. So we think with CAR T cells, we should just do all the work within that existing system. And so we call it point of care processing, doing the work where you can actually be caring for the patient. And by setting this up, we think we can not only help control costs, but we'll also be creating jobs and keeping jobs in the country. These experts that know how to do the engineering and everything, why wouldn't we want to be paying our own people to do that and creating the sort of high-tech economy that is good for us? It also will allow us to foster innovation. I've shown you, we've got our own ideas for how to make a better car. Why wait for Novartis to think of these things? We've got ideas. Let's make sure that we're able to put our ideas out there in the real world. So to make this a reality, we've created the Canadian CAR Tcell program. And we're doing this, it's us in BC doing this in collaboration with a group in Ottawa at the Ottawa Health Research Institute. And we pulled together a bunch of scientists, doctors, nurses, health economists, and created a big team to tackle this. And our first trial which we hope will open about a year from now, will be basically trying to recapitulate the results I talked to you about tackling these leukemias. And the way it's going to work, our collaboration, my colleague Rob Holt in Vancouver has designed the CAR that we're going to use. Our colleagues in Ottawa are going to actually make the vector that's used to do the engineering, the genetic engineering. They've got a facility that they can use to make these things. We've got clinicians in Vancouver and Ottawa who will be collecting the T-cells from the patients. The T-cells will then be flown here to Victoria to our lab across the parking lot, and our job will be the factory for this first trial to actually do the engineering of the T-cells in our clean room that I showed you. And then we'll ship the T-cells to Vancouver and Ottawa to be actually given to the patients to complete the whole thing. And then, of course, there will be medical teams in Vancouver and Ottawa that will take care of the patients and make sure that they will deal with any side effects and send them home in good shape. So this is what we put together. Now, it's complicated for a first step. In some ways, it's maybe like Novartis in that we've got just one factory so far. But our goal is actually, this is just proof of concept to just show that we can do this. And we're trying to do it in a way that we could then replicate it in cities across Canada. So Calgary, Winnipeg, you name it, could join our network and put together their own factories and do their own part of this. But we didn't want to, it's too hard to start with too many cities involved, so we just picked 2 initially to get it off the ground. And to give you an example of how we're trying to really think ahead, but how could you really do this in a cost-effective way at every major city in Canada? We're even though we've got this nice clean room, we'd like a way of doing this that doesn't require a $3 million clean room to build. So there's a product that's come out just in the last few years that actually is like a bread maker, to use an analogy, that actually will do the genetic engineering of the T-cells all in one unit that would basically fit on this podium. And so you basically put your T-cells, your engineering vector, some various ingredients and stuff in this thing. You push a start button and come back 10 days later when the little bell rings and your CAR T-cells are ready to give to the patient. So this is about a $250,000 instrument. So you pay a little bit up front, but it then can do this manufacturing in an automated fashion. So we think this is what the future is going to look like. Not factories in New Jersey, but little machines like this in the major hospitals across Canada. So we're going to prove that that can work. And if you come on the tour, we've got this in-house now, and you can see it and see more about it. So I just want to end by, I might have lost some of you along the way when I started talking about genetic engineering and how crazy is this guy and who let him in, and I'm sure he's got degrees, but doesn't seem to be part of the real world. Well, the reality is, this is where the world is in 2017. We are now genetically engineering cells and using them to treat cancer. And it might sound complicated now, but I think we're kind of with this in this business where computing was back in the 1970s. Yes, it's complicated. This is what computers used to look like. And skeptics in those days said, couldn't imagine that there would ever be a computer in everybody's home, let alone a computer now on something the size of a wristwatch. Another example is with gaming. If we think of video games today, and I think we can all remember, I remember certainly, this could have been me, it's not, but video games in the late 1970s, that's what we had. And think of what we have now with virtual reality and so on and so forth. So with all this CAR T-cells, as fancy as it may sound, really this is where we're at. It's just generation 1, and there's so much that lies ahead, and that's why I'm excited about this field. And if you still need convincing, I'll just end with a quote from Terry Fox. about that we can all do the impossible, and that's, but I think actually the impossible is now looking possible. Yeah.
So would there ever be a possibility that T-cells could be donated the way that organs are now donated? Like, is that something that is even in the realm of what scientists are thinking about? Or
Yeah,
just
absolutely, yeah. So the way things are currently done, as you picked up, it's the patient's own T cells that we're using. You'd think it'd be ideal if you could just have an off-the-shelf, grab a bottle of T cells and give them to the patient, and not have to do this manufacturing for every patient. And that may come. There's a number of groups working on what they call universal T cells. The question is, will they work as well as the patient's own T cells? Because if they don't, the part about manufacturing for every patient is not actually the hard part. So I don't think that's necessarily the biggest problem we need to solve. The bigger problem is to actually get this working against other cancers. But that, I think what you're saying is maybe where we'll be 20 or 30 years from now. Yeah.
What I don't understand is why isn't Merck or Pfizer or Bristol Myers here saying to you, here's $2 billion, you develop it and give us the right that when it's done, we will manufacture it. I mean, these are big multinational drug companies that have to be looking at this saying, man, if we can solve this issue, we can sell the drugs. Or are they doing that? Or are they doing their own people locked in their own labs down in the States someplace? Carol, what you're doing?
Sorry, asking, has Merck given me $2 billion? So the
Pfizer
answer
put $1 billion into R&D without batting an eye. I mean,
Sure,
they're
yeah.
huge.
yeah, no, you're right. I'm obviously being facetious.
And they must be excited about what you're doing and the results you're getting and your.
Well, to be fair, and I blurred the line between what I'm doing and what I'm taking credit for. I'm not meaning to take credit for that.
Yeah.
All the CAR T-cell stuff, we've not done any of that yet. We're gearing up to do it. So those breakthroughs came from Israel and the US. But how did Novartis end up being a leader now on the commercial side? They bought the technology from the University of Pennsylvania. So the inventions actually happened in universities. And this is how the model works, this is how drug companies work. They have their own in-house scientists, but they're not dumb. You know, if they see a breakthrough happening at a university, they'll go buy it. And that's, so that could happen here. We could come up.
So they'll monitor what you're doing, and if they see progress, they can come in and buy it from the
cancer agency, in my case, exactly, yeah. So therefore, we put patent protections on things so that
Yeah.
if somebody did want to buy it, it's actually, without a patent, it's worthless. So we do put those things in place.
Do you own the patent? Like, who owns the patent?
The BC Cancer Agency owns the patent for anything that its employees
I've
develop.
heard there's a danger when the pharmaceuticals get hold of a patent, they may never produce it.
Yeah, there's a risk that a great idea can be bought by a company, and they just put it on the shelf because they don't want it to compete with their other product. Often, though, in these kind of agreements, there's provisions for that sort of thing, that they need to actually advance it or else surrender it.
That
So
would be in the patent.
it wouldn't be in the patent, it'd be in the royalty or the licensing agreement. You would have some sort of clause around that. Yeah. Our thought, what if we came up, what if the MATES strategy I talked about, which we own, and I say is better than CARs, becomes a winner? That could be $1 billion market for sure. And so I think about this, how do I not become part of the problem then? Because if Novartis, picking on them, but if they bought it and then started charging $1 million for it, how's that helped the people in BC who actually contributed to allow us to do this research? So what we're talking about now is what's called a non-exclusive license, which is we could sell a technology to a company with the proviso that it needs to be free for use in Canada, for example, so that we can continue to use the technology for free to treat Canadian patients, and they could then go and sell it around in other countries around the world, for example. So that's one way to, I think, to deal with that ethical side of things. No, I want to be part of the solution, not part of the problem.
Yes, as I understand what you're telling us is that, I don't understand why you would want to sell it to start with. That's the first question. The second question is, to protect Canadians, you're saying if you do a licensing agreement, so that Canadians could use this, free would be the wrong word, but at a reasonable cost.
Yeah.
For hospital.
Yeah.
And that, I guess, brings me to my ultimate question, is that would you not then have people from all over the world coming to Canada and utilizing our so-called free medicine?
Right, they, except they can't. You can't just come into Canada and get medical treatment.
Well, that's not quite true.
Well, there may be exceptions, but in general, that's the rule. We don't allow what's called medical tourism.
You don't.
No, our system, it's not me or cancer agency, our whole nationwide healthcare system does not, you can't buy your way into our healthcare system. If you're a Saudi prince, for example, you could go to the Mayo Clinic and have whatever they're offering, but you can't come to Canada. No.
But what about my first question, and why are you even considering selling the rights to our paid-for inventions?
Well, it's a fair question. I would want to see, as the inventor of something great, I would want to see it used around the world. And yet, we're not in a position, with our current operation at least, to produce these kind of products for and distribute them around the world. That would mean creating our own enterprise that could do that. So that would either be a non-profit or a for-profit company. It would no longer be something a hospital like ours or clinic would be engaged in. So by selling it, and the proper term is licensing it, because it's a...
So it's altruism is what you're talking about. So
Well, that-
India or whoever could utilize what we develop.
Yeah, but it's not all just altruism. There would then be a revenue stream, a licensing fee that comes back to the cancer agency, and that then is used to help fuel more research and things. So that's the way our system works. We can't only work on donor dollars. When we have discoveries, if there's any monetary value to them, we do try and get that back. But it's put towards research. So that's the way the system works.
You started out talking about the immune system, right? That's the starting point here. And as I understand stem cell replacement theory, it's essentially, the layman's term, rebooting the immune system. Is there any stem cell research on the horizon for cancer?
Sure. Yeah, so stem cells are already used to treat leukemia. So when you have a bone marrow transplant, and this has been around since the 70s even, you're basically wiping out the immune system and replacing it with a healthy immune system.
But why wouldn't that work for other cancers, since all cancers have basically an immune system component, data component?
Yeah, well, it's a good question, and it's been tried, because it does seem, kind of logical that if it works for one cancer, it should work for the others. So during the 80s and 90s, there was a lot of work in trying to apply it to other cancers. It just doesn't work. And probably the main reason is you can reboot the immune system, but if the immune system, just like your computer when it's restarting, if the cancer's there, it's actually going to think that that's normal tissue. As it's kind of being rebooted, it is imprinting itself. It's learning what's normal, what's abnormal. And it assumes that anything it sees initially is normal. Again, because your immune system is trying to not attack healthy tissues, so it often errs on the side of caution. So you end up actually possibly in a worse circumstance when you've now got an immune system it has grown up with this tumor, and just thinks, oh, this is normal. Gentleman back, and then I see you over here.
Yeah, I imagine there's research, I mean, you're research trying to engineer the immune system to attack cancer cells, but I guess is there research that is just trying to figure out why cells become cancerous? Like, I mean, instead of trying to solve something that's been created, sort of trying to attack it from getting so it doesn't become, like, attacking it before it becomes a problem. Like, your research is directed at defensive move, something's happened, and you're countering it, but ways of why do cancers do, like, why do cells break down? Why do they break down and become cancerous? I mean, is there studies in some areas of the world where it doesn't happen, like diet, or I mean, what sort of things are causing this to happen?
Yeah, there's
if
lots
you
of
attack that instead of trying to attack what's already happened. You know, it's kind of like fighting a war. You're on the defensive of what's caused, but trying to go to prevent it from getting there. I imagine there must be stuff. Like
Yeah, that makes the most sense of all. An ounce of prevention is worth a pound of cure. I believe that
It
too.
seems like
So
some level of complexity.
there's three major approaches to controlling cancer. At least this is us in the field. This is how we divide it. There's prevention, so understanding how to prevent cancers from starting. There's early detection, which is finding them as early as possible when there's still those more or less innocuous bumps that aren't harmful. And the third is treatment. And in a perfect world, prevention would take care of everything. But I don't think that's ever going to be possible that we could prevent all cancers. We could prevent half of all cancers if we got tobacco out of our society. That is responsible for 50% of the total cancer burden in our society still. It's going down, fortunately, but not fast enough. The other things, since you've brought this on, you'd better be eating fresh fruits and vegetables. Sorry, that's still true. Exercising, maintaining a healthy body weight, avoiding too much sun, are the 5 big things we can all do. And if we all did that, the whole society would be much better off. But there's still aging, and just the longer you live, the more likely it is that something bad's going to happen. It's just probabilities. So each of us has about 10 to the 13th cells in our body. So 10 to the power 13, which is 10 trillion cells. So what are the chances that you can live 100 years with 10 trillion cells and something bad will never happen? You know, it's just sort of another way to look at it. It might sound a little callous, but that's sort of the basic statistics. And as the cells get older, they pick up these mutations, and there's not a lot we can do to stop it. So prevention, we could take us halfway to a world without cancer. But that other half, I would hope early detection would cover at least another quarter of the problem. Prostate Cancer Group, you would understand the challenges with that. We've got the PSA test, which sort of works. Works well enough that it's recommended for most people. We've got the digital rectal exam. I mean, how backward is that? But that's kind of our modern diagnostic method. And it's not because people haven't tried. It's really hard to find cancers early enough. You're basically looking for something the size of a wart in a person, and it could be buried deep inside them. And remember that until somebody has a particular type of cancer, until you become a prostate cancer patient, the day before that, nobody knew what kind of cancer you might get. So if we're really going to screen our population and find these cancers, we have to look for each of the 200 types of cancer in every person and hope to find it. So that's the challenge, and I'd never like to be negative and make it sound like, oh, nothing's possible. But that's the challenge. So maybe the next generation will figure out how to actually do that. It's really, really tough to screen. So I think for our lifetime and probably the next generation, treatments are going to be really important. I think you've been waiting a while to say.
How much coordination and cooperation is there between nations? Because our health system is literally the same as in the UK and Australia and in Europe. And, they have free medicine. Your facility here is relatively small compared, the global picture. So you think that there would be more interaction and communication, and this would be done through this academic model which you mentioned rather than the major pharmaceutical companies. Is this going on, and how valuable do you see it?
Yeah, there's a lot of international collaboration and cooperation, especially in the academic circles. So I travel a lot, more than I'd like to actually, but going to conferences all around the world and meeting with people to share ideas. I would give them the same secrets I told you tonight. I put my best ideas out there, even at the risk of somebody scooping me. I believe in just putting the information out there, and if somebody can take my idea and cure cancer faster with it than I did, then I'm happy. And I think most academics have that attitude. Of course, there's always a bit of competition. We've all got egos too. We want to, be the first, etc. But I think there's a pretty healthy dynamic there. I'm on the advisory board at the Mayo Clinic, for example, for their ovarian cancer program. So even though I'm from a small research center in Victoria, which we all know not everyone's heard of internationally, made enough of a reputation in the ovarian cancer world that I get invited to, without bragging, but to the top meetings and top places in the world, because we've got expertise here that other people don't have. So it's, it's a pretty distributed activity. We all publish in the same international journals as well, so we all read each other's papers, and we review each other's grants, and so
I think a more intense relationship, where you actually say, okay, we've got this problem, and we'll share this portion, you'll concentrate on this, and we'll do this part of it, to sort of have a wider field of exposure, if you like, that's coordinated.
Who's paying, which
Yeah.
province, in which country, which taxpayer?
well, so to
I'm
give
a cynic about that, as you can tell. Ontario has different standards than BC for healthcare and so forth.
Yeah, so you need to encourage these things to happen. They don't necessarily just happen spontaneously. But I'm the Terry Fox Research Institute. People do the Terry Fox Run. Where does that money go to? Something called the Terry Fox Research Institute. They are trying to create a network or strengthen the networks within Canada. I'm part of a project with Princess Margaret Hospital in Toronto, where they're for our adoptive T-cell therapy. They're paying us to play nicely with each other and share all of our secrets and work on protocols together and divide and conquer the problems. By paying us, what I mean is they're providing grant money for these kind of activities. I'm also part of, to give another example, the U.S. Army, believe it or not, is funding us to be part of a network of groups in North America, Europe, and Australia, looking at ovarian cancer survivors. Women who beat the odds and live 10 years or more are fairly rare, sadly, with that disease. So the U.S. Army said, we'd like to understand who are these long-term survivors and what are they doing right. And so they're investing millions in creating this international network. Because these patients are fairly rare, you need to get a lot of hospitals involved so that you can get big numbers of patients to study.
So I think
We're gonna have to break it up, but Brad Nelson's gonna stay for a 15-minute nutrition break, so if you have more questions, how about you could talk to him?
Yeah.
Is that?
Thank you.
okay?
Absolutely, yeah, thanks for,
Okay.
lost track of time. Thanks very much.